Hey guys, Miss Cote here, and here's the next episode of the Energy Unit. In the previous video, we did a brief introduction to cellular respiration. In this episode, we're going to start looking at the process of cellular respiration in more detail, as particularly the inputs and outputs at each stage. So let's get started. Last time we left off going through the three main stages of cellular respiration, and used this diagram as an overview of what's happening and where it all happens. You'll be seeing this diagram a lot during this chapter. Let's start at stage 1, glycolysis. Here we start with glucose and split it into two molecules of pyruvate. So we go from one 6-carbon molecule to two 3-carbon molecules. Glycolysis is a multi-step reaction. There are 10 separate reactions involved, all involving a different enzyme. These reactions can be split into two major phases. There's an energy investment phase where an initial input of energy is needed to get the reactions going. And the second phase is called the energy payoff phase, where we extract some of the energy from glucose and transfer it into usable forms. And one important thing to note, glycolysis will happen regardless of whether oxygen is present or not. So this is a process in both anaerobic and aerobic respiration. Remember that glycolysis takes place in the cytosol. So that means all organisms have the capacity to do glycolysis, which does make a little bit of ATP through substrate level phosphorylation. So here are the 10 reactions that take place during glycolysis. Once upon a time, before the redesign of the curriculum, you were expected to memorize the names of the molecules and enzymes at each step. Now you just need to understand an overview, basically what goes in and what comes out. Although if given a diagram of all the steps, you should be able to interpret the diagram and be able to answer questions about what's happening. Here's the overview. We start with glucose and end with pyruvate. In order to start glycolysis, we need two molecules of ATP. That is our energy investment. However, our investment will pay off because we do end up with 4 molecules of ATP and 2 molecules of NADH at the end of glycolysis. Overall, you can remember it as the 2s, 2 pyruvates, 2 NADHs, and 2 ATPs. Even though we make 4 total ATP, you need to subtract the initial investment of 2 ATPs at the beginning of the pathway. And that takes care of glycolysis. We've extracted a little bit of ATP at this point, but most of the energy that's still available is still stored in the 2 molecules of pyruvate. So we still have some work to do. If there is no oxygen, then the pyruvate molecules will enter into an anaerobic respiration pathway, which we'll discuss in the next video. But we're going to keep going with what happens in the presence of oxygen, continuing on the aerobic respiration pathway. When oxygen is available, the pyruvate will now enter into the mitochondrion in eukaryotic cells for stage 2 of cellular respiration. If the cell is a bacterial cell that can do aerobic respiration, these reactions will still take place in the cytosol since bacteria don't have mitochondria. One thing though, pyruvate cannot enter the citric acid cycle as is. It needs to go through a series of 3 steps to be converted into a molecule called acetyl-CoA. These steps are referred to as either pyruvate oxidation or the link reaction, since it links glycolysis with the citric acid cycle. And these steps happen in the matrix of the mitochondrion. Let's look at what happens. The 3-carbon molecule of pyruvate enters into the mitochondrion through a transport protein. In 3 steps, a molecule of carbon dioxide is removed from pyruvate, a molecule of NAD+ is reduced to NADH and H+, and a molecule of coenzyme A is attached. This creates the molecule of acetyl-CoA, which is the molecule that can enter the citric acid cycle. Coenzyme A also makes the entire molecule very reactive, so it's a very short-lived molecule. It's looking to react with something. Also remember that for every molecule of glucose, there are 2 molecules of pyruvate, so this link reaction needs to happen twice, once for each pyruvate. Which brings us to the citric acid cycle, which is also called the Krebs cycle, named after Hans Krebs, the scientist who first worked out all the steps of the cycle. This part of cellular respiration is where the cell finishes breaking down and extracting the rest of the energy from the pyruvate molecules. At the end of the citric acid cycle, one molecule of pyruvate produces one molecule of ATP, 3 molecules of NADH, and 1 molecule of FADH2. But remember, there are 2 pyruvates per glucose molecule, so there will be 2 turns, and these numbers will have to be multiplied by 2. The citric acid cycle also takes place in the matrix of the mitochondrion. It consists of 8 steps, each with a specific enzyme. And like glycolysis, you don't need to memorize the steps and enzymes involved, but I want to just quickly go through the cycle with you. The acetyl-CoA, made in the link reaction, which has 2 carbons, joins with a molecule called oxaloacetate, which has 4 carbons. This reaction produces a 6-carbon molecule called citrate or citric acid, and this is why it's called the citric acid cycle. The next 7 steps decomposes the citric acid back down to oxaloacetate, basically to regenerate the cycle, producing ATP and the energy carriers along the way. So citrate is converted to isocitrate, which is an isomer, and then oxidized to alpha-ketoglutarate. How do you know that this is oxidized? Hopefully you notice that during this step, NAD+ is reduced to NADH. So if something is reduced, then something else is being oxidized. It also loses a carbon as carbon dioxide. So now we have a 5-carbon molecule. In step 4, alpha-ketoglutarate is converted to succinyl-CoA. Another molecule of carbon dioxide is lost, so the molecule is down to 4 carbons, and another molecule of NADH is produced. A molecule of coenzyme A is also attached to the molecule, which makes for an unstable bond. Remember, the CoA is very reactive. The CoA is replaced very quickly by a phosphate group, which immediately gets transferred to GTP, which is transferred to ATP. I know, it's complicated, but in the end it makes a molecule of ATP. Here, ATP is created through substrate-level phosphorylation, and the new molecule is a 4-carbon compound called succinate. In step 6, succinate is oxidized to fumarate, and the electron acceptor is a molecule called FAD, which is a relative of NAD+. FAD will become reduced to FADH2. Step 7 rearranges the bonds with the help of a water molecule to make malate, and one more round of oxidation and the reduction of NADH+ to NAD+ to NADH to regenerate oxaloacetate. Now the cycle is ready to begin again. Remember, for one glucose molecule, this cycle has to go around 2 times to completely break down the 2 molecules of pyruvate made in glycolysis. So while you don't have to memorize any of these steps, it's important to know what the purpose is and what the inputs and outputs are for each stage. Remember that one of the products of cellular respiration is carbon dioxide. Here is where the carbon dioxide is produced. And while we've finished getting every bit of energy that we can get out of the original glucose molecule, where did the energy go? We've made 1 molecule of ATP, 4 molecules of NADH, including one from the link reaction, and 1 molecule of FADH2 for 1 pyruvate. So in total, for 1 molecule of glucose, which will be 2 molecules of pyruvate, we double these numbers to get 2 ATP, 8 NADH, and 2 FADH2. We can use the ATP directly for energy, but now we need to see what happens to the NADH and FADH2 molecules. At this point, we've only made 4 total molecules of ATP from glycolysis and the Krebs cycle. Most of the energy is still sitting in the energy carriers, which are NADH and FADH2. In order to convert the energy into ATP, those carrier molecules need to be taken to the last stage of cellular respiration, which is oxidative phosphorylation. In eukaryotes, oxidative phosphorylation takes place in the inner membrane, or the cristae, of the mitochondrion. In bacteria that can do aerobic respiration, this takes place in the plasma membrane. Oxidative phosphorylation can be separated into 2 parts, the electron transport chain and chemiosmosis. The electron transport chain is where NADH and FADH2 drop off their high-energy electrons to complexes of proteins embedded in the inner membrane. Notice they drop off the electrons at different points in the chain. And when they're finished, they've been oxidized back to NAD and FAD, and they can go back to glycolysis or Krebs and be reduced again. As the electrons get passed from complex to complex, and you don't need to know the names, they give off the energy they've been storing. Notice that there's no ATP in this picture. ATP is not being made at this point. Instead, the energy that's being given off is harnessed to pump protons, the H pluses, across the membrane. This is creating a proton gradient, which we'll discuss in a second. And when the electrons reach the end of the electron transport chain, they combine with two protons and oxygen to make a water molecule. This is why oxygen is so vital to cellular respiration. It acts as the final electron acceptor. When there's no oxygen, the electrons have nowhere to go. And here is where the water is produced. Remember, this is one of the products of cellular respiration. Wow, that was a lot of information from this one picture. So let's finish this by talking about chemiosmosis. So all of this energy is now in the form of potential energy in the proton gradient. The electron transport chain has created a situation where there are more electrons in the intermembrane space than in the matrix. Those protons are just waiting for the chance to get back into the matrix. This is done by a special channel protein called ATP synthase. It ends in ASE, so it's an enzyme. A synthase refers to making something, and it starts with ATP. So ATP synthase is the enzyme responsible for making ATP. I believe I've mentioned in an earlier episode that these enzymes are usually named for the job that they do, and sometimes it's easy to figure it out. ATP synthase, when opened, will allow the protons to flow from an area of high concentration to an area of low concentration. And the energy being given off from that, it's an exergonic reaction, is being used to make the ATP. Using the gradient to create ATP is called chemiosmosis. And this is what ATP synthase looks like. It's actually pretty cool in how it works. As the protons flow through the channel, it causes the protein to spin and ultimately drive the reaction to convert ADP in an inorganic phosphate into ATP. Going back to the big picture, the energy taken out of glucose is temporarily stored in the energy carriers, NADH and FADH2. They transfer the energy into the electron transport chain, which is used to pump protons through active transport from the matrix into the intermembrane space, creating a difference of charges or a gradient. This is called a proton motive force. The protons are allowed to diffuse back into the matrix through ATP synthase, which creates ATP. And so at this point, the cell has accomplished extracting energy from food and converting it to ATP. Now the cell can do some work. There's always been discussion about exactly how much energy is produced from one molecule of glucose. The model we have in our book, which we're looking at here, has glucose making about 30 to 32 ATP. But just be aware that different models may show a slight difference in the number of ATP being produced. When I was in school, I learned that every molecule of NADH was responsible for making 3 ATP, and every molecule of FADH2 was responsible for making 2 ATP. The main reason NADH made more than FADH2 was that NADH put their electrons into the chain at an earlier position. Scientists are now arguing that it may not be that high. Also, energy is required to move the molecules into the mitochondrion to finish cellular respiration. In any case, if there is ever a multiple choice question about the amount of ATP made from one molecule of glucose, the correct answer will be somewhere in the 30s. About 34% of the energy from glucose is converted to ATP. The rest is given off as heat. While this doesn't sound very efficient, it actually is. It's more efficient than the engine in a car, which only converts about 25% of the gasoline it burns. Just one more reason why cells are pretty amazing. And that about sums it up for this episode. Stay tuned for the next episode where we'll wrap up this cellular respiration thing by looking at anaerobic respiration. What happens when there's no oxygen available? So write down any questions you have to ask in class and be a good human.